Section 19.1, Population Demographics and Dynamics. By the end of this section, you will be able to describe how ecologists measure population size and density, describe 3 different patterns of population distribution, use life tables to calculate mortality rates, and describe the 3 types of survivorship curves and relate them to specific populations. Populations are dynamic entities. Their size and composition fluctuate in response to numerous factors, including seasonal and yearly changes in the environment, natural disasters such as forest fires and volcanic eruptions, and competition for resources between and within species. The statistical study of populations is called demography, a set of mathematical tools designed to describe populations and investigate how they change. Many of these tools were actually designed to study human populations. For example, life tables, which detail the life expectancy of individuals within a population, were initially developed by life insurance companies to set insurance rates. In fact, while the term demographics is sometimes assumed to mean a study of human populations, all living populations can be studied using this approach. Population size and density. Populations are characterized by their population size, the total number of individuals, and their population density, the number of individuals per unit area. A population may have a large number of individuals that are distributed densely or sparsely. There are also populations with small numbers of individuals that may be dense or very sparsely distributed in a local area. Population size can affect potential for adaptation because it affects the amount of genetic variation present in the population. Density can have effects on interactions within a population, such as competition for food and the ability of individuals to find a mate. Smaller organisms tend to be more densely distributed than larger organisms. Figure 19.2. Australian mammals show a typical inverse relationship between population density and body size. Estimating population size. The most accurate way to determine population size is to count all of the individuals within the area. However, this method is usually not logistically or economically feasible, especially when studying large areas. Thus, scientists usually study populations by sampling a representative portion of each habitat and use this sample to make inferences about the population as a whole. The methods used to sample populations to determine their size and density are typically tailored to the characteristics of the organism being studied. For immobile organisms, such as plants, or for very small and slow-moving organisms, a quadrat may be used. A quadrat is a wood, plastic, or metal square that is randomly located on the ground and used to count the number of individuals that lie within its boundaries. To obtain an accurate count using this method, the square must be placed at random locations within the habitat enough times to produce an accurate estimate. This counting method will provide an estimate of both population size and density. The number and size of quadrat samples depends on the type of organisms and the nature of their distribution. For smaller mobile organisms, such as mammals, a technique called mark and recapture is often used. This method involves making a sample of captured animals in some way and releasing them back into the environment to mix with the rest of the population. Then, a new sample is captured, and scientists determine how many of the marked animals are in the new sample. This method assumes that the larger the population, the lower the percentage of marked organisms that will be recaptured, since they will have mixed with more unmarked individuals. For example, if 80 field mice are captured, marked, and released into the forest, then a second trapping 100 field mice are captured and 20 of them are marked. The population size, n, can be determined using the following equation. The number of marked first catch times the total number from the second catch divided by the number marked during the second catch is equal to the population size, n. So using our example, the population size would be 400. 80 marked from the first catch times 100 captured in the second catch divided by 20 seen marked in the second catch. 80 times 100 divided by 20 is equal to 400. These results give us an estimate of 400 total individuals in the original population. The true number usually will be a bit different from this because of chance errors and possible bias caused by the sampling methods. Species distribution. In addition to measuring density, further information about a population can be obtained by looking at the distribution of the individuals throughout their range. A species distribution pattern is the distribution of individuals within a habitat at a particular point in time. Broad categories of patterns are used to describe them. Individuals within a population can be distributed at random, in groups, or equally spaced apart, more or less. These are known as random, clumped, and uniform distribution patterns, respectively. Different distributions reflect important aspects of the biology of the species. They also affect the mathematical methods required to estimate population sizes. An example of random distribution occurs with dandelion and other plants that have wind-dispersed seeds that germinate wherever they happen to fall in favorable environments. A clumped distribution may be seen in plants that drop their seeds straight to the ground, such as oak trees. It can also be seen in animals that live in social groups, schools of fish or herds of elephants. Uniform distribution is observed in plants that secrete substances inhibiting the growth of nearby individuals, such as the release of toxic chemicals by sage plants. It is also seen in territorial animal species, such as penguins, that maintain a defined territory for nesting. The territorial defensive behaviors of each individual create a regular pattern of distribution of similar-sized territories and individuals within those territories. Thus, the distribution of the individuals within a population provides more information about how they interact with each other than does a simple density measurement. Just as low-density species might have more difficulty finding a mate, solitary species with a random distribution might have a similar difficulty when compared to social species clumped together in groups. Figure 19.3. Species may have a random, clumped, or uniform distribution. Plants, such as dandelions, in A, with wind-dispersed seeds, tend to be randomly distributed. Animals such as elephants, in B, travel in groups that exhibit a clumped distribution. And territorial birds, such as the penguins shown in C, tend to have a uniform distribution. Demography. While population size and density describe a population at one particular point in time, scientists must use demography to study the dynamics of a population. Demography is the statistical study of a population changes over time: birth rates, death rates, and life expectancies. These population characteristics are often displayed in a life table. Life tables. Life tables provide important information about the life history of an organism and the life expectancy of individuals at each age. They are modeled after actuarial tables used by the insurance industry for estimating human life expectancy. Life tables may include the probability of each age group dying before their next birthday, the percentage of surviving individuals dying at a particular age interval, their mortality rate, and their life expectancy at each interval. An example of a life table is shown in Table 19.1 from a study of Dall mountain sheep, a species native to northwestern North America. Notice that the population is divided into age intervals, column A. Their mortality rate per 1,000, shown in column D, is based on the number of individuals dying during the age interval, column B, divided by the number of individuals surviving at the beginning of the interval, which is column C, multiplied by 1,000. The mortality rate is calculated by mortality rate equals number of individuals dying divided by the number of individuals surviving times 1,000. For example, between ages 3 and 4, 12 individuals die out of the 776 that were remaining from the original 1,000 sheep. This number is then multiplied by 1,000 to give the mortality rate per 1,000. So in that example, the mortality rate is equal to 12 divided by 776 times 1,000, which is approximately 15.5. As can be seen from the mortality rate data, column D, a high death rate occurred when the sheep were between 6 months and a year old, and then increased even more from 8 to 12 years old, after which there were few survivors. The data indicate that if a sheep in this population were to survive to age 1, it could be expected to live another 7.7 years on average, as shown by the life expectancy numbers in column E. Table 19.1. This life table of Ovis dolly shows the number of deaths, the number of survivors, the mortality rate, and life expectancy at each age interval for Dall mountain sheep. Survivorship curves. Another tool used by population ecologists is a survivorship curve, which is a graph of the number of individuals surviving at each age interval versus time. These curves allow us to compare the life histories of different populations. There are 3 types of survivorship curves. In a type 1 curve, mortality is low in the early and middle years and occurs mostly in older individuals. Organisms exhibiting a type 1 survivorship typically produce few offspring and provide good care to the offspring, increasing the likelihood of their survival. Humans and most mammals exhibit a type 1 survivorship curve. In type 2 curves, mortality is relatively constant throughout the entire lifespan, and mortality is equally likely to occur at any point in the lifespan. Many bird populations provide examples of an intermediate or type 2 survivorship curve. In type 3 survivorship curves, early ages experience the highest mortality rate with much lower mortality rates for organisms that make it to advanced years. Type 3 organisms typically produce large numbers of offspring, but provide very little or no care for them. Trees and marine invertebrates exhibit a type 3 survivorship curve because very few of these organisms survive their younger years. But those that do make it to an old age are more likely to survive for a relatively long period of time. Figure 19.4. Survivorship curves show the distribution of individuals in a population according to age. Humans and most mammals have a type 1 survivorship curve because death primarily occurs in the older years. Birds have a type 2 survivorship curve, as death at any age is equally probable. Trees have a type 3 survivorship curve because very few survive the younger years, but after a certain age, individuals are much more likely to survive.